Well, hello there. Welcome to Agronomy Day virtually. I am Alex Lipka. I am an associate professor in the Department of Crop Sciences. And today I'm going to give a really brief overview of my lab's research program. So if I am able to not mess anything up on Zoom, let me go ahead and share my screen. Okay, so here's my presentation. So play from start. Okay, so I am a statistician by training, but I do a lot of work in quantitative genetics in plants. And my field is very somewhat, jargon specific. So I have the added challenge of explaining why my research is important. So why don't I go ahead and do this? So um, what I envision the Lipka Lab is fitting into research and breeding is as follows. So important strides have been made in the past decade, or more than the past decade, in coming up with really accurate genotype data. So, you can come up with DNA markers that tags your favorite genes of interest that can be used in plant breeding. In addition, strides have been made in obtaining high throughput phenotype data, such as drones taking pictures of crops and obtaining a whole bunch of useful phenotypes. So what I envision my lab doing in my research is statistical G2P analyses. So what we do is we develop statistical models that connect the genotype to the phenotype, and this can help us make better decisions and help ensure that all of the monetary and time investments that we're making in these high throughput genotyping and phenotyping technology lead to meaningful results in the field, and importantly, accelerate the breeding cycle to help face the challenges that we have ahead of us in the next couple of decades. So to be clear, what we do at the University of Illinois is we use statistical models that associate the genotype to the phenotype. Our basic statistical model is this. We have an equation. We have a y variable and we have an x variable. Our y variables are trait of interest, and our x variables are one or more genomic markers. And so these models seek to accurately model the relationship between the genotype and phenotype, and to develop more accurate genomic selection models, which I will talk about in a second. But briefly, genomic selection is utilizing the entire genome information to predict phenotypes, and that can substantially speed up the breeding cycle. So some ramifications of the statistical models is that we can identify genes. We can characterize the genetic makeup of agronomically important traits. And we can develop more flexible statistical models that can analyze a wider range of traits, be it survival, be it discrete traits such as lodging and not lodging. And as I already briefly mentioned, these statistical models can help us reduce breeding cycles. And so that's why we go into this jargon, and that's why we do all this stuff. So talking about jargon, I'm going to talk about one example of a statistical model that incorporates a not often tapped resource of genetic variability. And what this is epistasis. Briefly, epistasis is the interaction between loci. And so since I am here on Agronomy Day and a lot of you work in crops, I'm going to talk about dogs, okay? So let's say that we have a phenotype, fur color of Labradors, ranging from black to brown to yellow. And let's pretend that we have two different genes or two different loci, singular version of loci is locus. So let's say we have the B locus controls black versus brown, and let's say that the E genetic locus controls yellow versus not yellow. So in this picture up here, we have all possible 16 combinations of genotypes between the E and the B locus. And then superimposed on each of these 16 rectangles is the fur color. So what we see is that no matter what the B locus is, whether you get the capital B or the lowercase b genotype, if you have little e genotype of the E locus, your color of your fur is going to be yellow. So thus, the expression of what you get from the B locus is dependent upon the E locus. This is an example of epistasis. So this is how we quantify epistasis in a statistical model. Don't worry, there's not going to be a pop quiz on this model, but we just want to open the hood and see what's going on behind the scenes. Like I said a couple of slides ago, our Y variable is our phenotype or trait of interest. So then we undergo a statistical selection procedure to determine a set of markers that have an individual non-zero effect on our genes, on our trait. And then also we pick pairs of markers that multiply with each other and have an interaction term which contributes to the variability of our trait. These, two-way interaction terms is our epistasis, okay? And then we also have our statistically important grand mean and random error term. And so then we can do a statistical procedure to determine these significant single markers or pairs of markers that have a major say in variability of your Y variable. And so we incorporated this statistical model into some software, which we decided to call SPAEML. So if you see the word SPAEML in the next couple of slides, we're referring to this model. So what we did in a recently published paper was we took the statistical model for a test drive using simulation data, and we compared it to existing approaches that do not do as good of a job theoretically as accounting for epistasis. So what we see in this graph is that the red lines and the blue lines and the red bars and the blue bars all approximately look the same. So this means that SPAEML does a good job, as good of a job as competing approaches, at least, for detecting, epistasis. And moreover, what this graph is saying is that, so we see that whenever we look at the maze data set, which is in our whenever we look at our human data set, which is our first and third rows, we see that the blue line goes up to the top as our x-axis increases, or our blue line is at the top on the third, or our blue bar is at the top. So what this means, and also we have this particular situation where we have an epistatic and a non-epistatic signal at the same particular locus. So what all of this jargon means is this. We are able to quantify these intricate epistatic relationships between genotypes and phenotypes, and we are able to, and it is possible to use this for marker-assisted selection. So that is, we can use this model to identify particular pairs of genes whose interaction effects are important. We can use them for marker-assisted selection that can potentially substantially accelerate the breeding cycle for particular traits. Next thing I want to talk about is genomic selection. I talked about it briefly, but I just want to go into a little bit of greater detail. So what we start off with is a training population of, say, crops. And these have been genotyped with a genome-wide set of markers, as well as phenotyped with a trait of interest. So we'll go ahead and use our statistical procedure to fit a genomic selection model where your Y variable is your trait of interest, and all of your markers throughout the genome are your X variables. So armed with this trained genomic selection model, we can then go into some crop breeding material that we're interested in that have been genotyped with these markers, plug those markers into this genomic selection model, obtain predicted trait values, and we can make predictions based off of these predicted trait values. And so the reason why genomic selection is important is that we can select, make selections out in the field or for crops without having to phenotype them, without having to, wait a field season, or maybe for perennial crops, wait multiple field seasons to obtain your phenotypes. Thus, genomic selection can substantially speed up breeding cycles. So there are many different variations of genomic selection models that are used in practice. In my experience, most produce approximately the same results. So here is one of the models that's widely used. So we have the RR-BLUP model. Our Y variables are trait of interest. We have all of our markers included in the model as X variables. And then we have our error term and intercept. And then we have this penalty over here, which, helps us deal with some negative statistical ramifications and be able to use this model in a typical data set. So we have this penalty, and what it does is an advantage of this penalty is it enables us to fit our models, just like I said, but it might grossly underestimate the effects of really important genes. So what we looked at in the research I'm about to show is whether or not it translates into suboptimal performance in genomic selection models. So what we did was we compared the predictive ability of the model that I showed in the previous page, as well as a typical model where we augment it with additional X variables that account for those artifact genes across an extreme range of simulated traits. So what we saw is basically, for most situations, when we include these X variables tagging large genes effects, we actually saw a decrease in prediction accuracy. And that's exactly what this graph is showing us. Our x-axis depicts the number of fixed effects, the number of additional X variables included in our model. And then the y-axis depicts our increase in prediction accuracy over the traditional model. So we see that, this dotted line over here is at a y-coordinate of 0. Most of the dots are at or below 0. So what this is telling us is that for most of our traits, including these additional X variables, tagging these artifact genes actually decrease prediction accuracy. And this is an important result because prior to this paper, there have only been positive results reported on the practice of including, these artifact genes as X variables in genomic selection model. So what these results are showing is that, including these artifact genes are not a guarantee for increasing prediction accuracy. So that's what this means for plant breeding. This approach is not guaranteed to yield optimal prediction accuracies. And depending upon the trait, using traditional genomic selection models might be better. And this, I'll finally leave on a philosophical point. One thing that I've figured out, through my PhD in statistics and my research in quantitative genetics is that there's no one-size-fits-all statistical approach. The answer of what is the best statistical approach is always, it depends. It depends upon your data. It depends upon the field you are in. So it's really important to dig into your data, dig into your field season, and do some preliminary analyses. And that's, what the Lipka Lab is here for, to help make educated decisions about the optimal statistical analyses and how to implement it into your crop plant breeding season. Okay, well, that concludes my talk. Thank you for your attention, and I hope you enjoy the rest of your day at the virtual agronomy day. All right, bye-bye.